All right. Hello, everyone. I am Dr. Munlock. Welcome to today's talk. I will dive into biosimilars, which the presentation is titled Biosimilar Development and Formulary Management. I appreciate you being here. I'm coming at you from Manchester University in Fort Wayne, Indiana. And if you are worried that I'm going to talk about confidence intervals at all, I will. So it'll come, but we'll have a good time. Here are some objectives. The objectives are kind of big concepts. We'll look at roles of biologics and biosimilars in therapeutics, and then also where formulary management fits into all of that. And I do want to mention I do not have any conflicts of interest to disclose. Biologics, what are they? Now, in school, I could wrap my mind around what is considered small molecules. In the middle of the picture here, we have a generic antidepressant structure and the different receptors that it can bind to. This is typically how we're introduced to new medications and drug classes, especially in pharmacology courses. And granted, this is still a pretty overwhelming picture, but at least we have kind of a simple structure. It binds to different receptors and then causes an effect. Now, granted, I'm taking that as kind of a simplified view, but we'll take it a step farther when we go into biologics. Now, with those small molecules, we can fit them into different functional areas, like we can call them pharmacophores, where we can make specific changes to different areas of the structure. So we can alter them by adding different functional groups. And by functional groups, I mean elements or combination of elements such as an OH group here, maybe a CH3 group there, a bonded double O. Now, with the graphic here, we can take a relatively simple structure like morphine and then add these functional groups to different spots. So we could take morphine and make it into hydrocodone or oxycodone. Just some examples there. And that's going to affect the bonding. Again, we're kind of looking at like a 2D um, chemical structure, and then we can see what changes can be made. Granted, still complicated, but somewhat of a simplified view. And then we'll kind of take it a step farther and more of a maybe like a 3D realm when we talk about biologics. Now, with biologics, we have a little bitty aspirin on the left, and then we have typical examples of biologics moving towards the right with insulin, erythropoietin, and monoclonal antibodies as examples. And I feel like monoclonal antibodies come up quite a bit as examples of biologics. And then those numbers are Daltons, which is typically how we measure the size of a molecule. Now you can see the monoclonal antibody is over 100 times bigger or larger than our typical aspirin molecule. And I feel like, moving through school and just trying to understand biologics, it just kind of was hard to kind of wrap my mind around the large molecules. I prefer my molecules to be less than 500 Daltons, which is sort of like a Lipinski of five joke, but we'll keep going. So, biologics, again, what are they? We already established that they are large and complex molecules. They are derived from living organisms or from recombinant DNA derived medicinal products. There are quite a few examples that fall under the biologic umbrella, which I have some listed on the slide. Some of these may seem intuitive, like you may have seen them before, such as vaccines could be an example, insulin and monoclonal antibody examples. But we can also add certain hormones, enzymes, cell therapy, and blood factors to the biologic family as well. And then again, we have another picture with a monoclonal antibody with the different binding groups, the FC and Fab regions, and then we kind of have the microscopic view of a little structure that actually might be, if I could take a guess, that actually might be a aspirin again next to it. So yeah, these molecules are huge compared to the typical medications that we typically talk about or think about or learn. Now back to small molecules for a second. There are plenty of items to note here, but let me go into kind of the dichotomy we have with biologics compared to small molecules. I mentioned they are derived from living organisms, but the biologics, the structure as the building blocks are typically amino acids, which are also the building blocks of proteins. The structures are very heterogeneous, as they are typically a mix of thousands of amino acids, and that heterogeneous nature makes it kind of hard to characterize, with the structure typically, like I said, being kind of like a building block of proteins. There can be a lot of variability when it comes to stability, and with proteins, we know that they can be sensitive to light and temperature and enzymes. A big reason why most biologics can't be taken by mouth is that in your stomach you have enzymes known as proteases. So as soon as you would ingest a biologic, or a large molecule, which is mostly just protein and amino acids, the proteases in your stomach would break apart the amino chain so that you wouldn't actually be able to get a clinical effect from that, because your stomach enzymes would just break it up. So that's why most biologics cannot be given orally, and we see a lot of them given as an injection or an infusion. And obviously there's things to take into account with that, because patients might not always be comfortable with taking a drug by injection or by infusion. They typically like the slightly more easy oral route. And then even cost is going to come into play. Is this a drug where they need to come into the hospital or clinic and get an infusion? That's going to be nursing time and administration time. That's going to be a cost. So quite a few things to take into account there. And then immunogenicity, that's the possibility of having an immune reaction occurring, as they are from living organisms. That's a possibility that could happen. So a lot of these factors are needed to be taken into account by a specialist, which can make the prescribing complicated at times. But we'll keep going and keep dissecting biologics. Biologics are approved via the Biologics License Application, commonly abbreviated BLA, and the Biologics Price Competition and Innovation Act, or BPCI Act. This was designed to foster the development and approval of biosimilars, increasing competition, and then also potentially lowering the cost of biologic therapy as well. The complexity of these molecules, which we've touched on, allows for really specific targeting for therapeutic treatments. Despite the complexity of them, they play a large role in therapeutics. It seems like half the commercials on TV today highlight use of biologics in autoimmune or inflammatory disorders. I feel like most commercials nowadays are related to medications, and then, yeah, I think a decent amount of those you're gonna see the biologic. And a good way to recognize if something is a biologic is the very end of it. Actually, I'll talk about this again in just a second, but a monoclonal, if you want to know a drug is a monoclonal antibody, it's going to have MAB or MAB at the end of the name. So that's kind of a key there. And I'll point that out again as an example. But with these drugs, like I mentioned with the complexity, it's that very targeted nature and precise therapy. It provides more like kind of revolutionary ways to treat our patients, especially with reducing off-target effects, just because these drugs are very precise. They hit the receptor that they're supposed to. And sometimes we see a lot of off-target effects with maybe more commonly seen with small molecules. But yeah, different therapeutic treatment options, like I said, in autoimmune disorders, plenty of examples of oncology, endocrinology, gastroenterology, and neurology. So far in 2024, we've had four new approved drugs that were considered a biologic, and these are the trends from the last 9 years. I don't know exactly what I'd call that color, because obviously we have blue, but then there's kind of a light blue green. I could call it turquoise. I don't know. We'll talk about that later. But I would say I would say that's the smaller bar graphs are kind of like a turquoise um, color. That shows the amount of drugs per year that were biologics. So, for example, there were 50 drugs in total approved by the FDA in 2021, and then 14 of those were biologics. I did a CE in Nashville on those 50 drugs with my good friend and co-fellow Matthew Sherman back in 2022. And to note, it's that novel mechanism of action that allows biologic development for orphan disease states that may be considered maybe more rare. And then also on the slide, I have a few examples of the 4 new biologics so far approved in 2024 with their names and then their brand names as well. And I'll explain the naming conventions in just a little bit. So we talked about biologics, and I'll continue, but now I'll introduce the term biosimilars. And we can see here that there's constant approval per the FDA over the years and months. The very first was filgrastim back in March of 2015. And then we've already had approvals for biosimilars already in this year. So some very similar ones were that tocilizumab AAZG. Now, the biologic or first occurrence of a new biologic could also be referred to as the reference product. And most likely moving forward in this talk, I'll be referring to that first biologic as the reference product. But from there, we can talk about biosimilars of that reference product and see if that biosimilar would be considered an interchangeable biosimilar product. Now, I'll most likely say this again, but let me just say this now. Every interchangeable product is considered a biosimilar, but not every biosimilar is considered interchangeable. Now, what is a biosimilar? And even with this picture on the screen, we have the reference product or maybe the first occurrence of the biologic. Then moving down, we have the biosimilar and then interchangeable. And I'll say this again, but you can kind of think of a biosimilar like a generic, but I will also point out ways of why you can't make that. But in terms of simplicity's sake, you can somewhat make that relationship. Now, I know the naming can throw individuals off. I remember getting asked a drug information question on what the 4 letters after the name mean. And as I say that, do you know what those 4 letter suffixes mean? I'll let you think about that for just a moment. So they don't mean the answer is nothing. They are unique 4 letter suffix that are devoid of meaning. And notice the little part there where I mentioned assist with pharmacovigilance and to avoid confusion. Although I feel like that is rarely explained, the 4 letter suffix, because it kind of took me a while too to figure out exactly like, okay, that's just a way, kind of like a tag to show like, all right, we have the biologic product and now we have a biosimilar and it has this 4 letter suffix that doesn't mean anything. Now this is a made up drug, this replicanab, camab. I think I did okay with that. We can see that it's supposed to be a monoclonal antibody because of the MAB at the end. So the MAB at the end stands for monoclonal antibody. So it's a biologic, it's a monoclonal antibody. But right, those 4 letters, I feel like as pharmacists, we want to try to find meaning in everything. But I'll say it again, those 4 letter suffix has no meaning. It's just there to avoid confusion, which again, kind of helps, kind of doesn't, but there's some examples. And I do just want to point out, so I grabbed this example from the FDA, but with a reference product or like the first occurrence of a biologic, from playing around in the purple book, you don't typically see those 4 letter suffixes for the reference product. You typically see that thrown on when a biosimilar comes out. So it's kind of interesting that they actually have a little hypothetical 4 letter suffix for a reference product, but I think they just put that there for an example. Now, the biosimilar is sort of like a generic, although the biosimilar is similar to the reference product. And I would like to think that's where the name came from. Biologic, similar, biosimilar. I'm sure they had a lot of fun coming up with that name. So it's similar, but not identical. And by reference product, I mean the original biologic, like I said before. And then like a generic, so think of Tylenol versus acetaminophen. Tylenol is a brand name, acetaminophen is a generic. The biosimilar should have the same clinical results, should have no greater risk with repeated use, especially when compared to the original reference product. So they are similar, but not identical. And I'll point out reasons as to why we can extrapolate why it's not identical. So we'll get there. Quality control and data reviews are conducted for the biosimilar to make sure everything is safe and effective. The FDA defines a biosimilar as produced from the same source or living tissue, provide the same benefits when treating condition, and also the same strength is used when dosing. And also the expectation is that the biosimilar will have well, that the expectation is low in terms of a biosimilar causing new or worsening side effects. So we want it to be manufactured very similarly. When treating the condition, we want the same effects, and we want side effects to be low or definitely not worse. If you are familiar with the FDA approval process of small molecules, we see a similar stepwise approach for the development process of biosimilars, and we'll look at examples of studies too that are used when biosimilars need approval. Like I said before, these drugs need to prove similar safety and efficacy, but then also pharmacokinetics and pharmacodynamics will also play a role moving forward. So there's analytical studies, animal studies, which would be considered preclinical studies. We have clinical studies. At the end of the day, the FDA will review it, and then also there should be, at least in my opinion, some sort of post-marketing safety monitoring, or maybe pharmacovigilance studies, or sometimes those are even referred to as phase 4 studies. But we need to look at all these things in the developmental process to ensure safety and efficacy, which, like I said before, we have to do for small molecules, drugs like Tylenol and aspirin and all those kind of things, but we have to do that for biologics and biosimilars as well. To prove the biosimilar is highly similar to the biologic, there's comparative functional studies that are done. So certain factors are taken into account with these studies, and kind of moving down, I'll explain. So this expression system is where it's expected, the biosimilar is expected, or at least the proposed biosimilar product is expected to have the same primary amino acid sequence as its reference product. So there's these huge chains of amino acid, but it's expected to be the same as the reference. All steps in the manufacturing process for the proposed biosimilar should be established during product development. We also need to assess potential for detecting differences in quality between the biosimilars and the reference products, especially with different physiochemical properties. Functional activities or functional assays helps in the characterization of protein products. An analytical test should be performed to characterize the product in terms of their specific immunochemical properties. We need to characterize, identify, and then quantify impurities as well. Impurities happen in all sorts of manufacturing processes in small molecules and biologics, and those need to be taken into account. There should be assessment of the reference product, so there should be at least a base of information from which you develop the biosimilar product, and then justify reliance of certain, scientific knowledge about the reference product back to the biosimilar. Characterization studies should be performed on the finished product, and then finally, a comparison of the stability of the proposed biosimilar product with that of the reference product should be initiated. An example with that could be like force degradation studies. So a lot of things need to go into place, and the FDA advises all these in terms of making a biosimilar product after a biologic. I made the connection before between generics and biosimilars, but biosimilars, again, are not exact copies. We see from the previous slide all the effort done, but we can only get as close as possible. So a couple regulatory bodies take the amino acid sequence I mentioned that make up these biologics, in order to be identical. So the FDA, European Medicines Agency, Health Canada, they need to show the biologics need to be, you know, identical, but at least precise physiochemical and functional property need to be comparable. Although not all functional differences may need to be characterized, and then also impurities need to be taken into account. So a lot of testing, a lot of studies that need to be done, again, to ensure safety and efficacy. And luckily there's these regulatory bodies that help along with the process too. And I, have an example where biosimilars, or well, really just that picture, but yeah, biosimilars, kind of think of it like generics, but with a generic, that's an exact copy of the brand. With biosimilars, it's not 100% the same thing. It is similar. It's not exact, because then it would be called a bioexact, and those don't exist. Okay, circling back to the pharmacokinetic and pharmacodynamics that need to be assessed. Equivalency in pharmacokinetics may look at like common pharmacokinetic parameters such as Cmax, area under the curve, otherwise known as AUC, half-lives, and volume of distribution as well. Because that's how we're looking at how the body affects the drug. That's the pharmacokinetics. With pharmacodynamics, we may need to assess therapeutic parameters, like with the one example I have, like blood glucose changes with insulin. So pharmacokinetics assess how the drug affects the body. And just note, equivalent pharmacokinetics doesn't always mean equivalent pharmacodynamics and vice versa. And finally, they'll need to look at immune response. So that's where immunogenicity comes in again, too. So they need to look at a potential immune response for the biosimilar when comparing the biologic or reference product to the biosimilar. But now let's look at some studies real quick, just to kind of see what this looks like. So I feel like we see Humira quite a bit. This study treated these pharmacokinetic confidence interval margins. So here I am talking about confidence intervals. They treat these confidence interval margins kind of similarly to non-inferiority margins. Now, remember with non-inferiority, right? Everyone remembers non-inferiority, especially the non-inferiority margins, right? So as long, speaking of non-inferiority, as long as our confidence interval does not cross the non-inferiority margin, we can say our results are non-inferior. Now, I may have simplified that a little bit too much because you need to take into account if you're addressing safety or efficacy data, but for point of this talk, I'll leave it at that. With this example on the slide, as long as our confidence intervals, which are represented by the squares with the lines coming out of them, as long as those are not crossing our margins of 0.8 all the way to the left to 1.25 all the way to the right, they can say they are similar enough in terms of pharmacokinetic data, which in this case, they could. So again, they looked at pharmacokinetic parameters. They wanted to see if those margins were crossing. As long as they were within the margin or not crossing the margin, they were similar enough. It looks like they used 90% confidence intervals instead of 95, but I want to say that's totally fine. So just an example here, if they're looking at C max and looks like the confidence interval was 0.88 to 1.02. If the lower end of that confidence interval was 0.79, that would have been crossing that 0.8, so that would not have been good. Or if the upper end of that confidence interval was 1.3, that would be crossing the margin of 1.25. So again, we don't want it to cross those margins. Kind of like with confidence intervals, we don't want certain ones to be crossing 1 or 0. With non-inferiority, we don't want it to be crossing our non-inferiority margin. And then with now these biosimilar studies, we also want it to be kind of within this pre-specified margin. But I have another example on the next slide. With this study, I chose natalizumab because I think it's kind of a cool name. I wonder if it was named after someone named Natalie. But anyway, okay. So I think it's with this example, we mainly need to address figure A, where the confidence interval does not cross the lower margin of -2.1 or the upper margin of positive 2.1. So big takeaway here is that the reference biologic product and biosimilar product are similar enough. I would consider the last slide that we just looked at. We looked at pharmacokinetic data with Cmax and A1C, and then this one appears to be more of a pharmacodynamic data because we're looking at new active lesions, something clinical that's happening in in the patient. So we saw two examples of studies that need to be assessed for approval of biosimilars. And I believe that this study was done and helped to prove the safety and efficacy of a natalizumab biosimilar that, is out in the market. And then figure B is kind of looking more at a slightly more descriptive view of looks like new active lesions over 48 weeks, and looks like the reference product and then the biosimilar natalizumab were very, very similar. Those lines were very close to one another. But again, with figure A, that confidence interval, let's see, it's roughly going from like, you know, that negative 0.7 to somewhere in between positive 0.7 to positive 1.4, but it's not crossing that upper margin of positive 2.1, not crossing that lower margin of negative 2.1. So similar enough. Now I'll say this again, all interchangeable products are biosimilars, but not all biosimilars are interchangeable. Now, interchangeable biosimilars can be substituted by the pharmacist without the need of a new prescription. This substitution is similar to like a generic substitution that's done in pharmacies all the time. Although with these interchangeables, they need to be labeled as an interchangeable biosimilar, and they undergo strict FDA approval processes. And we'll look at that in just a moment. Like I mentioned, it can be interchangeable biosimilar. It can be substituted at the pharmacy level, but then needs to meet additional standards to be shown that it can be interchangeable. I remember when insulin glargine YFGN came out, and at the time I didn't know the difference between an interchangeable and biosimilar, or I didn't know the difference between an interchangeable biosimilar and then just a biosimilar. I I thought biosimilars were already around for a while, but this insulin glargine YFGN was the first interchangeable biosimilar. And then the next few slides will go into the nuances between the differences. So there's a lot of, I think again, the kind of the whole reason why I'm even doing this talk is I think there's a lot of confusion around biologics, biosimilars, and then interchangeable biosimilars. And then, of course, you got the four-letter suffix that is devoid of meaning, and it's just it takes a little bit to kind of wrap your mind around all of it. In 2010, the Affordable Care Act allowed abbreviated approval, leading to biosimilar and interchangeables. We already talked about the approval process of biosimilars, or at least when approval process of biosimilars when compared to biologics, but there are different additional points from the FDA, such as the comparison of the biosimilar to reference product. But we'll also see a stepwise approach through like I said, proposed comparison between reference and the biologic. It's fair to mention that additional post-marketing should occur by monitoring safety and effectiveness, and that is important. And then I'll also introduce a term that should help a little bit to distinguish between interchangeable and biosimilar. But these, in terms of looking into new interchangeables, those are looked at a case-by-case basis. There's oversight by the FDA, and we'll look a little bit at those cases as well. And we'll look at an example of a study that they do to approve an interchangeable. So guidance from the FDA, these interchangeable products can be switched at the pharmacy, although the switching needs to be proven to be safe and effective, just like with generics and biosimilars, like I mentioned. The clinical effects need to be the same as seen in the reference product, which is totally fair, at least I think is fair. And, we'd want to see that with generics as well. You may have had a patient that's has said that they can only have a certain brand, and there may be some validity to that. You know, different brands and generics may have different fillers and different diluents. Those can vary. But just like with generics, these biosimilars and interchangeables need to have a similar effect on the body and from the body. So remember, that's why they look at pharmacokinetics and pharmacodynamics. Sometimes it's some, they utilize different studies. They could utilize a pharmacokinetics study, they could utilize pharmacodynamics, but again, that's you can't always, some may vary, and it's not always vice versa. um but we need to assess when switching is safe, because like I said, with an interchangeable, that's done at the pharmacy level. So maybe a patient's on insulin glargine and a pharmacist wants to switch them to that new insulin glargine interchangeable. We, there was studies done to show that the switching should be safe. And again, that switching should be safe and the products should be comparable. And we'll see kind of an example or a diagram of what a switching study could look like. So here we go. So with the switching study, there's a lead-in period, then the switching period, and then you have some pharmacokinetic or maybe like an endpoint sampling that's done to make sure that the reference product and the interchangeable is comparable. So switching studies can occur to see if the reference product is comparable, which I've said before. Now think of the reference product as the brand name. So during the study, there's a switching arm and a non-switching arm, somewhat similar to a control, where the study needs to assess if the endpoints they're measuring is consistent between, like I said before, between using the reference and using the interchangeable. There's a washout period right before sampling, which is necessary, especially if there is variability in the half-life between the reference and interchangeable products. We see washout periods with other types of clinical studies as well, too. So some people maybe get the reference, some people maybe get the interchangeable, then the reference, then back to the interchangeable, and the measurements and endpoint sampling that's done at the very end wants to show, again, consistent and comparable results between the reference and the interchangeable. I wonder how many times I've said interchangeable already. So here's an example of a label. So you will encounter biosimilars due to that four-letter suffix. And with the labeling, you'll see the asterisk and note in the package insert. I chose trastuzumab, as it's relatively common. Note here that they say that there's no clinically meaningful difference between the biosimilar product and the reference product. And we'll see that again in the purple book. Now, it seems like with the FDA, you'll more often see this biosimilar asterisk. I found some package inserts that actually had like an interchangeable asterisk and then had like a little interchangeable paragraph, but it seems like they're more leaning towards the consistency of just the biosimilar asterisk and then explaining what a biosimilar is. And this is where the purple book will help with this. So the purple book, I'd really want you to kind of play around with it. You can put different biologics into the purple book, and it'll list which have biosimilars, which have interchangeables. I've actually run into a few that says that they don't have a biosimilar, but they do have an interchangeable. But like I mentioned before, all interchangeables are biosimilar. So I guess they're kind of saying that there's no biosimilar by itself that's not an interchangeable. But play around with it a bit. So, we have our reference product, we have our biosimilar that has no clinically meaningful difference, and then this is where this one quote that helps a little bit with where interchangeable, where there's no increased risk with switching, which is again done why there's the switching studies are done. But yeah, with the purple book, like I said, think back to the orange book. The orange book's used when we want to make generic substitutions. With the purple book, look to see what biologics are out there, and again, which biologics or which biosimilars would be considered interchangeable. So play around with that a little bit. So there's the insulin glargine, YFGN, Celtizo, which is an interchangeable for Humira. You'll be able to find that as well. And then they list package inserts. So I think those would all be good examples for you to play around with. Now, how do we handle all these biologics, biosimilars, and interchangeables? Typically, institutions have a P&T committee that can handle this. Although in the US, interchangeability needs to be addressed with guidance from the FDA and utilization from the Purple Book. Don't forget about the Purple Book. The FDA evaluates interchangeability using a step-by-step process that considers the safety record and, of course, the complexity of both the reference biologic and the biosimilar. Although prior to starting the necessary research, the FDA actually advises sponsors of biosimilars to get in early contact with one another in order to plan how to discuss and demonstrate interchangeability. Because again, we want to, we want these, if these drugs are helping people, we want them to be used, but then also another thing should be taken into account, and then other things should be cost. Cost always plays a role in healthcare where we can see billions of savings with generics. We can also see large savings in biosimilars compared to the reference product. So we can see biosimilars could be 15 to 35% cheaper than those reference ones. Not only will reduced costs help the institutions and clinics, but there'll be benefit to the patient as well, especially with less out-of-pocket pay for patients. Like I talked about a little while ago, a lot of these are injections and infusions, which is a whole other cost and thing to take into account. But at least if, we're treating the patient, we're, helping them, especially if it's like a rare orphan disease state, we want to get this, treatment and therapeutics to our patient. And then we'll continue to talk about formulary management. So when considering biosimilarity, the considerations that need to be made here, the issue of the proprietary production and not having access to the original cell lines are all valid reasons as to why biosimilars are not exact copies, right? So the manufacturing process has to be very similar. They have to have the same amino acid sequence. But again, these are large molecules and large proteins. Temperature, pH, and drift can all cause changes to efficacy. Drift is not that one Tokyo movie. Drift is differences that can affect the biologic's performance and safety profile over time. But even additional chemical processes that can happen that can affect the structure as well. Typically we see proteins and biologics folded in certain ways. We've talked about you've heard before with proteins, they could have like a primary string of amino acids, and then they become secondary and tertiary and quaternary, but then all these things can affect that, like oxidation and glycosylation and phosphorylation. Oh my, all those things can happen, which, like I said, these are very complex molecules, so they're not easy to duplicate 100%. So again, this slide is really just to show the complexity of it and why, it's our good old branded generic doesn't really apply to biologics. It's biologics and biosimilars, and then, even interchangeability comes into play as well. With that being said, here are some really big concepts that I'll dive into further with continuing slides after this. So these are all PT considerations. So the PT committee needs to assess what their institution is going to be using the biosimilars for, what data is already available, and then what kind of immune response could occur when switching products on that formulary. Because if it's a interchangeable, the pharmacist could switch it, but is that safe? Is that in the patient's best interest? In addition, I'll touch on the confidence of medical professionals when prescribing and switching biologics, where we know, some confusion can be taken into account, especially with the naming. Even when a product is used often and the prescribers are comfortable with it, another biosimilar or alternative biosimilar product may need to be added based on supply chain need and supply chain issues. Of course, cost will play a role. How does incorporating interchangeability going to affect workflow, such as like barcode scanning and labeling? And then where does reimbursement, cost is always going to come back. Where does reimbursement come into play? And again, education not only of the provider, but of the patient as well. So the next few slides will break up these concepts a bit more. Some clinical considerations should be addressed. We need to consider if it's being used on or off brand. It should be known, and I have a quote here, that the Potential exists for the biosimilar product to be licensed for more or more additional conditions of use for which the reference product is licensed. But of course, we want our formulary to be safe. We want our formulary to be safe and effective. So, in order to best evaluate safety and effectiveness, the committee may have to review other trials or other sets of data. Typical trials are our gold standard randomized control trials. We may have to look at additional or alternative trials to those. The FDA regulates biosimilar approval requirements, so they look at in vitro and in vivo assays to show the biosimilar's activity and potency, and just again making sure it's similar to the reference product without a clinically meaningful difference. There's studies on immunogenicity and then pharmacokinetics or pharmacodynamics, and that must demonstrate safety and purity and potency for all those approved uses, which we saw some examples. So the clinical studies needed depend on the residual uncertainty about biosimilars after all the functional and structural and animal studies, meaning actually clinical studies may often be limited, and clinical studies are very expensive to do. And finally, immunogenicity is going to be a factor. We talked about that. That's where switching to a biosimilar may cause an unwanted reaction. All kind of a heavy slide, but all things need to be taken into account, especially clinically when managing a formulary, and especially all things the P&T committee will have to look into. Now, with product considerations, most stakeholders prefer biosimilars to have generic names matching the branded biologics. However, unlike conventional generics, biosimilars structure or differ structurally and may not share the same generic name. Therefore, reasons for the confusion with naming comes from having multiple biosimilars, which is true, for each branded biologic having unique characteristics due to manufacturing. So using the same generic name could confuse prescribing, dispensing, reimbursement, and then post-marketing tracking. Now, if you ever use the FAERS database, FAERS adverse event, or FDA adverse event database, a lot of drugs are found in there under different names, and you want to take all those into account. Nuances like storage may play a role. Does it make sense to keep this product in your pharmacy? Where can you store it? For how long can you store it? All things to be taken into account. Now, but these biologics, I'm pointing out a lot of limitations, but these biologics can provide really great help to patients. Like I mentioned before, they have very targeted and precise mechanisms of action, especially when treating disease states. But we want to, with even that being said, we still want to avoid dispensing errors, labeling, barcode considerations. And, dispensing errors are a type of adverse event that we as healthcare providers can help prevent. So again, a lot of things to be taken into account clinically, but then also with the product as well. And then institutional considerations, touching a little bit here on the therapeutic interchange. So considerations for interchange include therapeutic and dose equivalents, efficacy and safety risks, cost benefits, clear interchange processes, making sure everyone's on the same page there, monitoring outcomes, monitoring efficacy. Payers may actually incentivize biosimilars to control costs, potentially actually raising co-payments for original biologics, but then the branded biologic or reference product manufacturers might actually offer discounts to encourage continued use of the original product. So that actually kind of poses a little bit of dilemma between cost savings and switching, because switching, like I said, can cause a potential immunogenicity reaction. I think I made that up, or I think I made up that term. An immune reaction, immunogenicity. So yeah, frequent switching can cause variable outcomes. Manufacturing changes can affect efficacy and safety. I talked about how, manufacturing these as a perfect clone is impossible. Even transitioning or switching between transitions of care should be taken into account. And then, at the end, pharmacovigilance data could help supplement the need for expensive clinical trials. Again, monitoring long-term how that patient's doing in terms of efficacy, safety. Are they on a new biosimilar, a new interchangeable? What's happening in terms of efficacy? What's happening in terms of immune effect? A lot of considerations there. So I brought up a lot, and these are all important questions here. So for the P&T committee. Not only will the initial review of the product, labeling, storage, regulating use of the biosimilars, switching, availability, all of that, not only will that all come into play, we need to consider education, pharmacovigilance, traceability, so like barcode scanning, nomenclature, so the naming, coding in the EHR, the electronic health records, pharmacoeconomic evaluations, bringing up cost and use and cost benefit, um, product considerations, so think PK pharmacokinetics, think PD pharmacodynamics, available data that's done that maybe can circumvent potential clinical trials, and manufacturing and supply chain. All are good questions here to be taken into consideration when adding and switching and utilizing these drugs for our patients on the formulary. Looking specifically, slightly switching gears, slightly not, looking specifically into the confusion I've mentioned at this point. So here's a survey here where 93 pharmacists submitted a survey focused on the naming conventions of biosimilars, where most of these pharmacists either worked in managed care or within an institution. Only 66.2% indicated familiarity regarding biosimilars, and 50.6 had familiarity with interchangeability. I don't mean to be biased by saying only, but as pharmacists, as the medication experts, these numbers need to be higher, or at least in my opinion, in terms of confidence with utilizing these. So, in terms of confidence with the graph on the left there, confidence in substituting interchangeable biosimilars was high when both products shared the same active ingredient or same non-proprietary name, although that confidence decreased when they did not share the same ingredient, or they did not share the same name, or when you throw in different prefixes and suffixes, that adds to the confusion, unfortunately. I don't think pharmacists like being told that something has no meaning. I can only imagine someone thought the letters and that the YFGN after insulin glargine stood for something. Maybe they thought it stood for your fasting glucose now. That was the one joke I had set up for this, but I'm gonna move on. Okay. And then finally here, cost. So we want to talk about the financial impact on the healthcare system, inpatient and administration costs, patient and institutional support programs, medical information technology, pharmacovigilance, drug shortages, monitoring treatment response in terms of efficacy. We want to look at additional monitoring that may be needed for therapeutic interchange and safety. We want to ask, will patient assistance programs be in place? Sometimes there's a lot of specialists that specifically help with prescribing and monitoring and changing biologics and biosimilars. Patient out-of-pocket costs will affect access and adherence. If it's too costly, they'll stop showing up or stop taking their medication. All things that affect all parties involved. So we're kind of ending this here with costs. Again, we want to always take our patients' safety and their disease state, their therapeutics into play. But then, yeah, these, like I said, these are very complex molecules with a lot of complexities to take into account. And this presentation kind of dives into a little bit of all of that. So let me ask a few questions. So here's a poll everywhere slide. Now, of course, poll everywhere is not working today, because that would make too much sense. You got to be really careful in academia not to make too much sense. So let me ask this question. Which of the following is not an example of a biologic? I'll give you just a minute or two to think about which of the following is not an example of a biologic. I think we've already seen filgrastim as an example. We've seen erythropoietin, but I'll give you just I wish this was working because then I could see your people logging in and giving their answers, but yeah, poll everywhere sick for the day. But that's okay. So the answer here is erlotinib. It definitely sounds like it could maybe potentially be some sort of biologic, but here's actually the structure of it. But yeah, if you would have asked me on the street, "Hey, this is biologic," just based on the name, I would lean towards yes. But yeah, here's a structure. That's not example of a biologic. The others were examples of biologic. Talking about Indiana, can pharmacists in Indiana substitute an interchangeable biologic? What are our thoughts here? I described what interchangeables were, but oh, sure. And I really probably should have said interchangeable biosimilar, but I don't think interchangeable biologic is necessarily wrong either. You could probably find it both ways, but interchangeable biosimilar is how I've been referring to it pretty much this whole time. So can pharmacists in Indiana make that substitution? The answer is yes. Normally, I would have a slide that has like a check mark, but again, pull everywhere is not working. But the answer is yes. Pharmacists in Indiana can substitute an interchangeable biologic biosimilar. But again, it has to be an interchangeable, and that's where the purple book will come into play. Look at the purple book, check to see if your biologic or biosimilar has an interchangeable, or if that biosimilar is an interchangeable biosimilar, then that substitution can occur. The studies that we looked at, last question here, or sorry, actually second to last question. The studies here for biosimilarity look similar to what kind of studies? Superiority studies, non-inferiority studies, or inferiority studies? I brought up the example of non-inferiority. That's how I mean, this is maybe a slightly more subjective question, but I think looking at these biosimilarity studies in terms of like a non-inferiority, like margin, I think that's similar. I believe I saw somewhere else where someone else pointed out the similarity, but I guess that's me also pointing out the similarity. So I thought, they looked more like non-inferiority studies, where non-inferiority studies were trying to say that a drug is not worse than something else. Kind of similar to what we're doing here. We're saying that the biosimilar is similar to the reference biologic that's available. Let me open up this. So what are your thoughts on formulary management after this talk, or what are your thoughts on prescribing? This is kind of where I'm opening it up to questions a little bit. If I don't get any questions in the chat, I will move on to closing thoughts. But let me just remain here for a moment just to see if anything comes up in the chat. You can use the chat or the Q&A box. I saw a comment by Indrani. Hi, Indrani. Thank you for being here. That's great. So I'll leave it here for just a minute. If thoughts or questions, or um, if you can think of any biologic or biosimilar jokes I can incorporate next time, happy to do that. But I'll remain here for just a moment longer. I think I have coffee left. I do. Okay, let me move to, there's no additional thoughts or questions. Let me move to my closing thoughts. Here we go. Closing thoughts. So in closing, biologics, biosimilars, interchangeables, all confusing things. Really, the big reason as to why I wanted to do this presentation and go into all this research is because before this presentation, I really didn't understand biologics and biosimilars and interchangeable. I didn't, I wasn't even totally sure what an interchangeable was, or I barely even saw that term before looking into this presentation. And I mean, there's a lot of really great presentations out there. I think, they look at biologics from kind of a 1,000-foot view. Obviously, you can take a really deep dive into each specific one and how they work and how they can affect patients. But hopefully, the angle that I kind of took with this in terms of looking at the development, but then also how formulary committees and us as prescribers and medical professionals, how we can, make substitutions, how our changes and interventions impact the patients. There's plenty to say with cost. A lot of these products are still, maybe would consider to be brand. So, cost is always going to be an issue there. But, as I saw with that graph that I made with the different drugs that are being approved every year. I mean, I would say roughly on average, this is just a Dr. Munlock quote, but I'd say roughly on average, we get maybe close to 50 drugs approved every year by the FDA. And then a decent amount of those, what was it, like 14, 15, just about, are biologics. So those are never going away. So I think kind of increasing our knowledge, our confidence, especially with like, all right, what is a biosimilar compared to a biologic? And then now you throw interchangeable in the way, and it's like, all right, well, do I have the confidence to use the purple book and say, all right, this is an interchangeable. I can I can substitute this for another product. What's going to happen if that switching occurs? Is there going to be an immune reaction that's going to occur? How confident do I feel that this is the same product and that the patient will do well? And I think, just because these are all so kind of new within the last few decades, I guess I didn't quite look up when the first, well, I believe insulin is technically considered the first biologic. I could be wrong with that, but that was my thought. But with, you know, monoclonal antibodies, which we see a ton, those haven't been around forever. And I think, they're new and we just need to become more comfortable with it. And at the end of the day, it's all about the patient and us treating the patient and making sure the patient is being treated for effectiveness and for safety, and that we're reducing possible adverse events and dispensing errors on their part. So it is complicated stuff, but I really enjoyed putting this together and presenting today. But those are my closing thoughts. These are my references, and this is my closing slide. And yeah, thank you for IPA for hosting this and having it. And yeah, let me know if there's any additional. What now, is that going to show my face if I just did that? I don't know.
It did.
Oh,
Thanks for presenting, Austin. It was a great presentation.
thank you so much. Thanks for having me.
For those of you who want to claim CE, the CE claim code used in Lecture Panda is AM as in Austin Munlock 24 for 2024, and B as in boy, D as in David, AM 24BD.